there. You can like cut and paste them and stick them places and all that kind of stuff. Okay, dudes, here's what we're going to do. Today, we're about to start getting into the really hairy physiology. It'll start on Wednesday. You're supposed to have a test on Monday. I'm not sure. I may move it back to Wednesday so you have a little bit more time for absorption. We did the sodium potassium pump last time, right? But we're going to sort of depend on you understanding that today. So instead of assuming that you understand it, I'm going to let you guys build it for me. Starting with concentration gradients, then putting in the channels, and then putting in the pump. And then I'm going to add to it because there's a bit of information that I only briefly touched on last time. So let's talk about the sodium potassium pump, okay? So this time you guys are going to help me do it. All right, so we're going to start with just the basics. FYI, if you have a question, somebody else has it too. And the stuff that we are doing today is fundamental to the really hard stuff that we're going to do on Wednesday. So I need you to ask a question when you get confused. I promise I'll be nice. If I'm not, it's certainly not about you. It's just my personality, right? You're like, I think it's charming when I'm snarky. Do you not? Okay. So we'll start with ECF, ICF. And the first question is asking you the things that you know, which is, will you please tell me where the sodium is high generally? No. Extracellular fluid, high sodium concentration. And it's relatively lower on the intracellular fluid, okay? And where is potassium high? Yeah, it's relatively high in the ICF and relatively low in the ECF. Okay, the numbers we'll get in there in just a minute because they're not super important quite yet. They will be. This is homeostatic for the cell. Let's tell a story in which you start here and then you put in your leak channels, right? And this is going to be a sodium leak channel. And this is going to be a potassium leak channel. And two questions. And I know that you have an asterisk when you call it a sodium leak channel because it just prefers sodium, but let's just say it's a sodium leak channel. Okay, which direction is sodium going to leak through this leak channel and why? Inside, because why? Concentration gradient. Okay. And same question for potassium. Which direction is potassium going to leak and why? Okay. And why? Concentration gradient. But now what I have to do is I have to give you a little bit more information and then build a little bit more stuff before I put in the sodium potassium pump. You ready? Okay, so concentration gradients, let's be a little bit more specific about it. The potassium concentration gradient is about 30 potassiums inside to every 1 outside. That's just comparing it to itself, okay? And with sodium, it's about 15 outside to every 1 inside. Okay, so now I have to tell you one other little piece of information, and then I have to tell you that we're eventually going to get a charge out of this story. Okay, so now looking at the concentration gradients for sodium and potassium, can you tell me, based on those concentration gradients, when I start to let them diffuse, which of the ions is going to diffuse more quickly? Potassium, because it's got a steeper concentration gradient. In addition, let me add one more piece of information to that to confirm what he just said, and that is, in addition to the concentration gradient, there are also more potassium leak channels. Okay? So now we can pretty much guarantee that potassium is initially going to leak faster. Does that make sense to you? Okay? More leak channels. Your textbook describes it as the cell membrane is initially more permeable to potassium because of the more leak channels and the higher concentration gradient. Is everybody okay with that? Okay, so now what I want you to know is, now what we are going to do is we are going to imagine the birth of a cell. It's a little bit of a fib, but it's a story that will make it all make sense. At the birth of a cell, let's imagine that the intracellular fluid has a net neutral charge, okay? For every positive, there's a negative, right? And the extracellular fluid has a net neutral charge, yeah? And then I stick in my leak channels. You with me? Okay, and then let it leak for a couple of seconds. Okay, With me? Stick in the leak channels. Started out neutral, neutral. Stick in the leak channels and let it leak for a couple of seconds. When I let it leak for a couple of seconds, you guys just tell me that which ion is going to leak faster initially? Potassium. So now I am going to let potassium leak out, sodium leak in, but they're not going to leak at the same rate. Yeah? Give it a couple of seconds, and where will I start to accumulate a positive charge?
Outside.
Yes. I will start to accumulate a positive charge on the outside. Okay. So let's think about that for a second. Somebody explain in your words how the heck that just happened. Go ahead.
The ratio of potassium on ICF versus ECF is 30 to 1, right? And sodium from outside to inside is 15 to 1.
Right.
So more potassium is going to leak out because the ratio is bigger for potassium.
Right. And there's more channels, right? That too. Okay. So what will happen on the inside if it started out electrically neutral and you let more positives leak outside than positives leak inside? It will get a slight negative charge. That good? Okay. Did I accumulate that negative charge, or did I just reveal a negative charge that was already present? Did I actually move any negatives? No. So there were negatives in there. I didn't tell you what they were yet. They're mostly proteins. Big negatively charged, like structural proteins and enzymes. We usually have a slight negative charge too. Is that okay? So I revealed the presence of negative charges. Okay, is that okay? So what I now have is this is homeostatic for the cell. So I told you before that the concentration gradients were homeostatic. Now I'm going to tell you that this charge difference is homeostatic too. Okay? And now what I'm going to have to do is to try to maintain or repair or put that back every time I jack with it, or maintain it forever. And so now what I'm going to do is I'm going to put the pump back in and think about the ratios at which it pumps. Okay? In order to maintain these concentration gradients, which direction do I need to be pumping sodium? Outside. Yes? Got it? Okay. Which direction do I need to be pumping potassium? Inside. And of course, to get things to move and dance their concentration gradient, I have to use ATP. But now I want to talk about the ratios, because the ratios can now start to make sense if you will let them. I told you guys that I call this the 3-2-1 sodium-potassium pump. It is 3-2-1 sodium-potassium pump because now that I have charges, I have two reasons for each of those ions to move. Okay? So I only don't have a charge for like an instant at the birth of the cell, and then for the rest of the cell's life I have this charge or trying to get back to it. Does that make sense? And now that I have the charge, one of these ions is going to speed up its diffusion rate. Which one? Now that I have this charge, which ion is going to speed up its diffusion rate? Sodium. Why? Now it's going down its concentration.
It's going down its
electrical gradient. Does that make sense? Electrochemical gradient. And what's going to happen to potassium? It's going to slow down. Does that make sense? Why? So I see some of you guys getting it, and I see some of you not. Go ahead.
It's a positive cell moving out to a positive environment.
Positive moving toward a positive. Doesn't want to go that direction. Positive moving toward a negative. Does want to go that direction. Okay? So now, for the rest of the life of the cell, because there's only an instant at which you didn't have a charge, which ion is going to move faster? Sodium is going to move faster now for the rest of the life of the cell than potassium, because it's moving toward its opposite charge and potassium is moving toward its like charge. Now let's go back to the sodium-potassium pump and say, why the hell does it pump in the ratios that it pumps? It pumps 3 sodiums out, 2 potassiums in, uses 1 ATP for the pump, because what it is doing is for the rest of the life of the cell, it is maintaining this concentration gradient despite the fact that the ions are diffusing down their, not just concentration gradient, but electrochemical gradient. Yeah? Does that make sense? Go ahead.
So when is the pump added in then? Is it always there?
So this is the story that I kind of tell you that's a bit. Okay, so here's what happens. All that shit happens at the same time. But I don't know how to tell that story to make it make sense. So you wouldn't have had the concentration gradient without the pump in the first place. Does that make sense? right? So where does the pump belong? Beginning, middle, or end? Yes. So the way that I tell it is just so that you can understand how the charge actually accumulates,
So
right?
there's not something that makes the pump come in?
No, so you basically have pump, channels, and concentration gradients all relatively simultaneously, but I don't know how to make that story make sense. Just like stick them all in at the same time. So sequentially, how I do it and how most textbooks do it is say, okay, we cannot acknowledge concentration gradient, yes. We can put in channels, yes, right? We can accumulate charge, yes. Then we can figure out what the pump does, yes. Realizing that it all actually happens simultaneously. Got it? Okay. This is homeostatic for the cell. Okay. Why do you care? You will care because in just a second we're about to use what we just built, which is a really, really dependable electrochemical gradient to start moving things without having to pay for them. Because can you see now that if I want anything to go in and sodium decides to just hold its hand, it can go in relatively readily, right? It's like, hey, you were already going to go in anyway, right? So that's what we're about to do. Does that make sense? So you'll start seeing today like why we did all of this. Okay, and then I'll tell you the sources of the negatives in just a little bit. Okay, so now this was all primary active transport. This is anyway. There are other pumps, FYI. You've got calcium pumps and chloride pumps and a bunch of different kinds of pumps. Really what a pump does is a carrier that moves something against its concentration gradient and uses energy. Okay, this is of course in every cell, so we talk about this one, but don't be surprised when we run into other ones later this semester. Actually today we'll run into one. So now the next type of transport we're going to do is called secondary active transport, and here's what secondary active transport does. Secondary active transport starts with the electrochemical gradient that you're already using, you already generated, and uses it to transport things without paying for them directly. So you're going to have a two-step process. The first step will not be active. No ATP is used. And the second step will be active. Okay? So secondary active transport, the first one does not require energy. So here we go. What we are going to try to do is to get glucose to move inside the cell without worrying about glucose's concentration gradient, meaning I don't care if there's already a bunch of glucose in the cell, I still want it. Right? And without paying for it directly. So the metaphor that I will use, and I always use for this, is you are in your car and you're about to drive to Florida. And you are paying for all your own damn gas. You've even got a place to stay. And you have somebody who says, hey, I want to go to Florida, but I don't want to drive and I don't want to pay for gas. So is she amazingly hot molecule like glucose? The answer is yes. Right? Because how much glucose do you want? How many hot girls do you want on the beach? All of them. How much glucose do you want in the cell? All of it, right? So what we are now going to do is, if sodium already has a mechanism of going inside the cell, we can use that driving force to bring things inside the cell for free-ish. It's almost free. Like, it's been paid for up front, and we're willing to like take the hit and take the hit later. So let's see what's going to happen. So what we are going to do is now, instead of just letting sodium diffuse into the cell, this time we are going to diffuse it, instead of through a channel, we're going to diffuse it through a carrier that has a binding site for glucose and sodium, and when both of them are bound, you use sodium's electrochemical gradient to move stuff into the cell. And it works because you worked really hard on sodium's electrochemical gradient. Does that make sense? Now, what if glucose was already high in the cell? We don't care, right? Now, we could get glucose to move into the cell by pumping it directly, but isn't that more expensive than doing this? Yeah? Okay, so I want to think about this. So what's going to happen, guys, is this is a carrier protein. Even though it's not showing it, I want you to imagine it's got a binding site for glucose and a binding site for sodium. Yeah? When they are both bound, then it will move glucose inside the cell. But if you do that 1, 2, 3 times, you're going to start messing up the sodium concentration gradient. But you know how to fix that, don't you? How do you fix it? The sodium-potassium pump. So that means, in theory, instead of pumping each glucose in, 1 glucose, 1 ATP, 1 glucose, 1 ATP, 1 glucose, 1 ATP. I get 3 of them in, 1, 2, 3, before I have to do anything to fix it. Do you see that that's way more efficient? Okay, so I'll show you an animation of this to sort of solidify it. You ready? Okay, so what I have is this is a carrier protein. It's a carrier protein for 2 reasons, guys. First off, it has to be a carrier protein in order to fit glucose, right? Yes? And second, it has to be a carrier protein to have shape-specific binding site at all. Yeah? So you're going to use a carrier, and I'm going to bring sodium and glucose in. Only if I do it a few times, it's going to start messing with my sodium concentration gradient. So what I'm going to have to do to fix that is I am going to have to pump it back out. Right? So now what you will see is you are going to start pumping it back out, getting rid of sodium. Does that make sense? Okay. So why is this called secondary active transport? The first step is really what? What is the first step? What kind of transport? Facilitated diffusion with a hitchhiker. Yeah? And then the second step is active transport, right? When you put the 2 of them together, they are secondary active transport. Does that make sense? Okay, so somebody explain to me in your words, why? Why am I doing this? What's the objective of secondary active transport? Go ahead.
Since you're already using energy to do one action, might as well.
Yeah,
Back off.
right, you like paid for it on the front end, right? So now you're going to get the benefit of bringing something inside the cell without paying for moving it against its concentration gradient, right? Does that make sense? So that's part of the reason we work so hard on this. It's not the only reason. We will see about 20 that we work so hard on this. But of course it wouldn't work if you hadn't generated this electrochemical gradient for sodium and been working on keeping it that way, right? So this is secondary active transport. Now, this is a really common mechanism of bringing both glucose, other sugars, and other nutrients in the cell, even if you already had a bunch of them. So what if I had a bunch of amino acids inside the cell, but I needed to do protein synthesis? Well, I need more amino acids inside the cell, and I don't want to pump them. So I'm going to move them in with a carrier protein that this time has a binding site for sodium and an amino acid, right? So a really common mechanism. You use sodium so frequently to get things inside the cell because you can see that it wants to come inside the cell. In addition, you can, by the way, also use it to flip things outside the cell like a revolving door. So watch. Bind sodium, but I want to get calcium outside the cell. And I use sodium's electrochemical gradient to flip something outside the cell. And I don't have to use ATP directly to get to do it. This time it just has a sodium binding site on that side and a calcium binding site on that side. When they both bind, it changes the shape of the protein, right? So this is called co-transport or symport when you're moving in the same direction, and counter-transport when you're moving in them the opposite direction, or antiport. Is that okay? Okay, so that's secondary active transport. Keep that in mind. We're going to use it again today. So the first step is not active, and the second step is. Examples, sodium is linked to co-transport of amino acids and sugars, and sodium is linked to counter-transport of ions. For instance, you can actually get H+ to leave the kidney by using sodium's electrochemical gradient without like pumping H+. Okay, so FYI, people get confused sometimes because now I've just told you 3 different mechanisms that I use to move sodium, right? So this was a leak channel. This was a pump. And then we just saw a carrier protein for facilitated diffusion. Are they all the same thing? No, they're 3 different ways of moving sodium. And not surprisingly, there are a bunch of ways of moving glucose too, because it's a pretty damn important molecule. So what I just showed you, instead of an insulin-dependent transport of glucose inside the cell, this one is insulin-independent, right? And you have it on some of your cells, like for instance, skeletal muscle cells and some of your neurons. You don't always want to be dependent on insulin. So there are a bunch of different mechanisms of getting things to move it. It's also going to be our absorption mechanism in the intestine, because we don't have insulin on the inside of the intestine, right? Does that make sense? Okay, All right, so now, for those of you who like to do organization of your knowledge, here is a summary table. You won't be able to memorize it by just looking at it. But this is a really good way to figure out whether you actually understand what you're talking about. So if you are doing a passive transport technique, what is it called? Is it using a protein? If it is it a carrier or a channel? Does it use energy? If it uses energy, are you using ATP or are you using electrochemical gradient? Does that make sense? So this makes you sort of be critical about your knowledge and figure out whether you can tell one type of transport from the other. So these are the 5 basic types of transport. And we're going to sort of quiz you on those guys in just a minute. Not a graded quiz, but another one. Okay. Now, the last big type of transport is the last one that we would choose. So remember our cheapest, fastest rule, right? So this type of transport is the slowest and the most expensive of all of them. So you would never do this if you could actually find another way to move something. So now what we are going to do is we're going to talk about vesicular transport, transporting things in vesicles, endo and exocytosis, basically, right? So these are really energetically expensive and they're way slower than the other one. Interestingly, even though they're super energetically expensive, we don't actually understand exactly how they use energy yet, meaning that we've never actually found where the hydrolysis is necessary to cause the endo or exocytosis, but we do know that if you remove ATP availability from a cell, it can't do endo or exocytosis. And we can see how much ATP it uses. We're just not quite sure what the hell is going on yet. So maybe somebody will figure it out before you guys are out of school. So really expensive. What they are going to do is they are going to move things in membrane-bound bubbles, not buggles, membrane-bound bubbles called vesicles. And remember that endocytosis is like this. So imagine that I'm a white blood cell, and here's my cell membrane. And here's a bacterium, a little coccus bacterium. And what I'm going to do is I am actually going to fold my cell membrane around it. And because of the phospholipid nature of the cell membrane and the fact that it's got fluid mosaic, when it joins, of course, it's going to form a bubble and then seal the cell back up, right? That makes sense? So that's the first one that you're seeing right there. So endocytosis, there's 3 different categories. Phagocytosis is when you take in big solids, like whole cells. White blood cells are famous for doing that. Pino or pinocytosis is when you take in fluids, so you'll fold around and get a bubble of extracellular fluid, right? And one of my students said it's easy to remember because Pinot is wine, right? So you're just like having yourself a drink. That's all you're doing, having yourself a drink. You can have Noir, you can have Grigio, I don't care what kind of Pinot you have. So you bring in something dissolved in fluid, and then the slowest of the endocytoses, is called receptor mediated endocytosis. And what this one is I want to bring in something that can't move by any other mechanism. There's some forms of iron that you just can't get in. There's a form of cholesterol that's got protein stuck on it, so it's not soluble, but you need to bring it into the cells. So receptor-mediated endocytosis is when you're looking for something super specific, like these purple triangles, but you need to wait until they're bound to receptors so that you know that you have them, and then activate endocytosis. So can you see that this one's probably going to be slower than the other 2, right? And do you want to bring it in when one thing is bound to a receptor? No, because it's expensive, right? So you're going to wait until they bind, however many, will trigger endocytosis, and then you'll do endocytosis, and you're going to bring this in, a bubble that has whatever you were looking for in it. Usually this is not what you do with waste products. This is something that you needed inside the cell. So this is called receptor-mediated endocytosis, and it's slower than Christmas, but sometimes it's necessary. Okay, and then exocytosis is how you get big stuff out of cells that can't use any of the other mechanisms. So I am insulin. I'm a pancreatic islet cell, and I just made, I'm a pancreatic islet cell, and I just made insulin, okay? Insulin's a relatively large protein, and I want to get it out. Why can't it diffuse straight through the cell membrane? Huge. Why can't it go through a channel? Too big. Why can't it go through a carrier?
As big as the carrier.
Usually big or bigger than the carrier, right? So how the hell do I get it out? Well, I have to because insulin doesn't do anybody any good if it stays in the pancreatic cells. So what I'm going to do is I'm going to make insulin and just put a little cell biology back together with this. Where would I have made it in the pancreatic islet cell? Which organelle? That's where I package it. Where do you make big old proteins? Endoplasmic reticulum. Then you package it in the Golgi and you put it in a little bubble, right? And then you take that bubble and you smash it up against the cell membrane. And since it's made of the same stuff of the cell membrane, right, it just forms and fuses. You don't even lose any phospholipids. And then it dumps the product outside, right? And you don't lose any phospholipids. So that is exocytosis. That's how you get big, huge stuff outside of cells. Good? But it's slow, too. So you would never, ever want to use that if you had another alternative. So that's exocytosis. Okay, so now, what I want to do is I want to look to see if you guys are really getting this. So instead of me telling you how things would travel, I want you guys to try to tell me how things would travel. I promise I'm going to be nice. And you have to defend it. Remember that if you are doing something that is more expensive or slower than a cheaper mechanism, you should be able to tell me why you didn't do the cheaper mechanism. Okay? You ready? So raise your hand if you can tell me how each of these substances would cross the cell membrane and why, what type of transport is it, and if it uses a protein, is it a carrier, a channel, or pump? Okay? So first one is oxygen is moving down its concentration gradient across the cell membrane. What kind of transport? Simple diffusion, I agree. Somebody raise their hand if they can tell me why you can get it across by simple diffusion. Go ahead.
Because it's a nonpolar molecule.
Nonpolar lipid-soluble molecule. Check. Got it. Go ahead.
It's pretty small. So
And it's small. Yes. Nonpolar lipid-soluble and small. Yes. Okay. Now, again, potassium going down its concentration gradient across a membrane. What kind of transport? Somebody else raise their hand. I'm not going to let Charles answer this one. I like you very much. Potassium is diffusing down its concentration gradient. It's going to be diffusion, but will it go?
It'll go through a channel.
Channel. Okay. Now, who can defend to me why you can't do simple diffusion straight through the membrane with potassium? Charge. Channel. Diffusion. Yes? Okay. Got it? Okay. Cool. All right. Next one. An amino acid moving into a cell for protein synthesis. Amino acid moving into a cell for protein synthesis. Go ahead. Raise your hand, Ashton. Carrier. Okay, Ashton, why couldn't you diffuse it straight through the cell membrane?
It's too big.
Too big. Why couldn't you use a channel? Still too big. So you're going to use a carrier. Okay, and diffusing through a carrier is called what kind of transport? Facilitated diffusion. Yes, good. Facilitated diffusion. Okay, all the rest of you people, you got to start talking, man, because you're going to wear these four out. Okay, go ahead.
Is there a way to identify how big it's going to be, or if it's going to be small enough?
Use the example. I'm always using examples that I've already given you. So I gave you amino acids and sugars as mid-sized water-soluble. I gave you oxygen as a lipid-soluble one. Ions are small and charged. So I'm going to try to, until you guys get really confident, use the same iterations of examples over and over again, right? And in addition, remember I showed you that if you could count the molecules, C6H12O6, or look at the size of an amino acid, it was going to be mid-sized. But proteins are a whole bunch of amino acids stuck together by peptide bonds. So those are generally big things. That makes sense? Okay, so now, next one. A large antibody protein secreted by a white blood cell. Go ahead. Other way. Exocytosis, exactly. Okay, so good. Exocytosis. So same damn question. Why can't you do simple diffusion? Too big, water soluble. Why can't you use a channel? Too big. Why can't you use a carrier? Too big. Biggest things that we're talking about are big proteins being secreted, right? So exocytosis is plan Z, but you don't have any other choices at this point, right? Okay. Water. I want you to sit here and think about this one. Okay? Water moving from a cell's ICF into a highly concentrated saline solution. I'm going to wait a second. I'll let you guys answer if nobody else does. I'm going to let them sit there and be uncomfortable until they're brave enough to raise their hand. Go ahead.
Is it simple just because the water is going to move where there's less concentration of it?
Of water.
Right.
But what do you call diffusion of water across a membrane?
Osmosis.
Yeah, so it is diffusion, guys. Osmosis is simply diffusion of water across a membrane, right? And it's moving from water's high concentration to water's low concentration. Does that make sense? So osmosis is just a special instance of diffusion. That's all it is. Last one, and I'll stop torturing you for now. Okay, calcium being pumped into the calcium-rich extracellular fluid to allow for muscle relaxation. Give it a shot. Go ahead.
Active transport.
Yes, active transport. Okay, but it's an ion. Why can't I diffuse it through the channel? Because what?
It has a charge and it's going against this concentration.
Yes. It's going against its concentration gradient. So regardless of size, regardless of whether it's big or small or ugly or pretty, in order to get it to go against the concentration gradient, you are going to have to use what kind of protein? Carrier protein plus ATP, which is a pump. Yeah? Carrier plus ATP. Got it? Okay. So these questions right here, these are phys questions that B students, A students should be able to get, right? Because instead of me telling you, it's you using the application and telling me, right? So I want you to keep working to the point that you start being confident about being able to answer new iterations of the same thing that you saw before. Like, I didn't tell you yet, but I can figure it out, right? So hopefully what you are hearing is that I really, really, really want you to be able to answer a question I haven't already told you the answer for, but gave you all the tools to answer. Does that make sense? Okay. Now, will your entire test be those questions? No, but I promise there'll be enough so that a student cannot make an A without thinking about novel situations. Okay, go ahead.
So for number 2, is that still called simple diffusion just through a channel, or is that called something
Yeah,
else?
well, usually when I diffuse something through a channel, I call it diffusion through a channel.
Okay.
Right? I don't care if you put simple diffusion through a channel, but just in my mind, when I think about simple diffusion, it's either going straight through the membrane or there's no membrane at all. And just to delineate, not that this is like an absolute truth with people, to delineate, I will say diffusion through a channel. Simply because like diffusion through a channel means it's going to be a little slower than simple diffusion, right? So there is like some difference. I'm not going to nitpick you about that terminology, right? I do kind of want you to know that it might be a little slower. Okay, you starting to apply it? Okay, now let's put it into a situation in the human body. Ready? Okay, so in order to get something into the human body, can we agree that it has to cross an epithelial membrane? Yes? Epithelial tissue. So now what we're going to do is we're going to take all this damn cell stuff and we're going to put it in the human body and we're going to try to figure out, if I drink water, how does it get into my bloodstream? If I eat salt, how does it get into my bloodstream? If I eat sugar, how does it get into my bloodstream? And what we're going to do is we're going to use these tools. Cheapest, fastest way, defend your position. Cheapest, fastest way, defend your position. You ready? Okay, first we have to remind you what an epithelial membrane is. Okay, and specifically the vast majority of absorption is going to go on across the epithelial tissue lining the small intestine. Yeah, that's where most of it's going to go on. Okay, so does anybody remember? I know a couple of you guys will. Does anybody remember what type of epithelial tissue is lining the small intestine? Come on. It is simple columnar with microvilli on the surface. Yeah, simple columnar. Why microvilli, by the way? Increased surface area for absorption, exactly. So here's our objective. We just ate or drank something, it's in the lumen of the small intestine. We need to get it all the way into the bloodstream, right? So that's where we want our stuff. And we are going to have to do transport here across the apical or apical surface, yeah? But then we're going to have to, of course, diffuse it through the cell. No problem there, it's tiny. Then we're going to have to do transport across the basal surface of the cell, through the interstitial fluid into the bloodstream. Luckily, the capillaries that are behind the intestine are mostly fenestrated, so if you get it close, it just goes straight through the hole. Like you don't have to worry about an extra transport mechanism there. So primarily what you're going to have to do is worry about here and here. Okay, you with me? So we are going to use the predictions that we have before. Fastest would be what kind of transport? Simple diffusion, and then next fastest? Channel. Then next fastest? Carrier. And then if you had to, you would pay for it with a pump. And now let's compare primary and secondary active transport. Which one is cheaper or faster? Secondary is cheaper than primary. It's also faster. Does that make sense? Okay, at least the part that does the transport is faster. Okay, so I want you guys to use those predictions. Here we go. So, first thing we're going to do is we're going to eat some chips, and we're going to get the sodium into our bloodstream. Why do we need it? Because here, that's why we need it, right? Because it is supposed to be in relatively high concentration in the extracellular fluid everywhere. And of course, we are constantly peeing it out and sweating it out, right? So we have to actually replace it. Luckily, the American diet has no shortage of sodium in the diet. I am one of those people who prefers salty, savory things over sweet things all the time. I would eat a pizza over a brownie any damn day. Okay, so here we go. First thing is, how do we get it to move into the apical surface of the cell? I'm not going to tell you. I'm going to let you sit there and argue it out in your head. How do we get it to move into the apical surface? Charles?
I'm looking at it. It looks like it's just a channel.
Okay, it does look like a channel, but you could also figure out that it's going to be a channel. Because you should know something about the intracellular concentration of sodium. What do you know? It's low. So I just ate chips. Yeah? Relatively low on the inside of the cell. It wants to move in. Can't move straight across the cell membrane, so it has to use channel. Yeah? The next cheapest fast thing. Yeah? Got it? Okay, so that's going to be a channel. Which means what type of transport is that? Diffusion. Diffusion. That's what I call it. Diffusion through a channel. Right? It's diffusion, but it's going through a channel. But you can say just say diffusion if you want to. I always say diffusion through a channel because it helps me separate it from simple diffusion. As long as you know what you're talking about, I'm not going to nitpick you. Okay? You with me? Okay, but I got it into the cell. Now I need to get it out of the cell and into the bloodstream. So now I need to get it out of the cell, guys. How do I move it out of the cell? I have to move it with the pump, but why do I need to pump it? You're going against its concentration gradient, right? So I'm going against its concentration gradient. It's high out there, low on the inside, so I have to pump it. Do I really care that it moves potassium at the same time? No, because potassium gets pumped in and then just diffuses straight back out. It's all right. There's loop channels for potassium. Does that make sense? And then once you get it actually into the interstitial fluid around the capillaries, it moves straight in because they're fenestrated. So you don't have to worry about that transport mechanism. Does that make sense to you guys? Okay. That protein at the bottom, this one right here, what kind of protein is this? It's a carrier, and when you add ATP to a carrier, it becomes a pump. Okay. Do you hear that it's important to me that you know that carriers are different than channels? Yeah. It will tank you on a bunch of questions if you don't get that down on the test. They are different than channels, okay? All right, so we just absorbed sodium from our chips, but now we are going to eat a chocolate chip cookie, and we are going to absorb the glucose. But we all know that chocolate chip cookies suck if you don't salt them, right? Do you guys not know this?
It makes it sweet.
Charles, have you never made homemade chocolate chip cookies from scratch before?
Oh, I was thinking that salt on top of that.
Oh, that's good too.
I'm like,
Flake salt on top of
it's
it?
like people saying.
Have you guys
salt
ever
on
eaten the cookies at
watermelon.
Caputo's? Have you ever been to Caputo's Deli? Oh, hell.
So it's like salted caramel.
Yeah, they have like
The sweet.
flake salt on the, oh my God, they're so good. Go get one today, you deserve it. You're gonna need it after this. Okay, so, yeah, in your sweets you need a little salt, or else they really do not taste very good. So what we are going to do is we're going to eat a chocolate chip cookie, and it's going to have both sugar and salt in it. Almost all of our food has salt in it. So what we are going to do is we're going to try to absorb that glucose. And we're going to try to absorb as much of the glucose as possible. Because how many hot girls do you want on the beach? All of them, right? How much glucose do you want in the body? All of it. What would you do if you had excess? Store it as glycogen, right? right? Store it for a rainy day. So we want to absorb all of the glucose if possible. So even though the intracellular fluid might already have a high concentration of glucose in it, I still need it to move in despite glucose's concentration gradient. So what is the most efficient way I can get it to move in without respect to its concentration gradient? Secondary active transport. So what I'm going to do is have a binding site for glucose, a binding site for sodium. Okay, I am going to move it in via what kind of protein? Carrier protein, yes. This is the first step of secondary active transport, which is basically facilitated diffusion with a buddy, the hitchhiker, right? That makes sense? Okay, now we got it inside the cell, but the cell does not get to keep all the hot girls just because they came to that beach first. You got to give it up. You got to share the love. So the cell has got to get the glucose out. So now let's talk about how you get the glucose out. We'll talk about the sodium in just a second. How do I get the glucose out? Well, let me tell you something important. This glucose right here never stays around for very long because it's got to go do its job, which is cellular respiration. Okay? It's got to do its cellular respiration job. So it's being carried away. Got it? So now I have high and low. So now you tell me how I'm going to get glucose to move out. Facilitated diffusion. Yeah? Why can't I use simple diffusion? Too big. What kind of protein is this? Carrier. Why can't I use a channel? Too big. Yeah? That makes sense? So I'm going to get it to move out, and as soon as it gets close to the fenestrated capillaries, it will move in. Right? Good. But then, of course, we have to acknowledge that we will create a sodium concentration gradient problem. Yeah? And how am I going to fix it? We're going to use the pump. Exactly. And we are going to have to pump sodium out to maintain the concentration gradient because we brought sodium, so much sodium in. Okay, so which one are your cells going to be using? Yes, both of them, right? So when would it bind? Whenever it could find those glucose transporters, then glucose is going to bind, right? Okay, the rest of the sodium will be moving in through channels, right? So it's just looking for binding sites. And of course, the more transporters you have, your better absorption rate, right? That makes sense? Okay, so what kind of protein is this again? Carrier. So we've got 3 carriers involved here. Yeah? A carrier, a carrier, a carrier. Good? Okay, everybody all right? Questions? Okay, last one. Easiest one, but they have crappy pictures of it. So, okay, so now I'm going to drink water, right? So I ate some chips, I'm going to drink some water, which is generally going to happen when you eat some chips. So now what are you going to do? So here's the deal. So watch with me for a second. So if I'm moving sodium, right, or any solute really, but we'll think about sodium mostly. If I'm moving sodium, I'm going to move sodium from the lumen to the intracellular fluid to the interstitial fluid to the blood plasma, yeah? And when I do that with sodium, do you see that I'm increasing sodium's concentration in all of those things sequentially? Yeah? And so what I am doing to water's concentration is what? Decreasing water's concentration in all those locations sequentially. So what I am doing is I am creating an osmotic gradient as I move solutes for water. Yeah? So what will happen is as solutes move, water will move, right? My phys professor said solutes suck, and sodium sucks the most, right? So you move sodium and you create a concentration gradient for water as you do that, right? So water moves in via your digestive tract by what process? Osmosis. It moves in by osmosis. And you guys know that you will, of course, move way more water in by osmosis if you eat a boatload of sodium, right? Have you ever just gone to town because there's like an entire tub of popcorn and somebody just left you with it at the movies, and how much do you eat? I ate the whole damn thing, and then what happened yesterday? I need new pants, and I'm apparently going to be married for the rest of my life because I can't get my wedding ring on, right? Okay? I mean, I know that was a plan anyway, but not like under duress. Just like, you can never leave! Okay. You ready?
I know if I want my wife to stay with me, I just basically increase her salt.
Yeah, you can increase her salt concentration. Of course, she could walk off with the ring, too.
Sure.
We're relatively mobile. Okay, so on your test, your next test, you will have to explain to me on the test the epithelial transport, which means movement across the epithelium, of glucose or sodium or water. One of the three. And I am not going to tell you which one, because if I told you which one, how many would you learn? One of them. So I'm going to tell you one of them will be on your test. And I don't care if you draw it or write it, but if you draw it, it better be labeled, because I am not a visual person. So label it like big and pretty with flashing things. Look here, dummy. So, all right, last one. This one is not in this chapter, but I think it goes in this chapter. They didn't agree with me, so I'm going to do it in this chapter anyway. So, filtration. Filtration is movement of water and small solutes across a membrane, usually an epithelial membrane. But it is pressure-driven, not concentration-driven. Okay? So, let me give you an example of filtration and then a metaphor for filtration. So here's an example of filtration. If you have two fluids, one of them is high pressure and one of them is low pressure, and you have a permeable membrane between them, then you're going to get filtration. So of course, in every single capillary in your body, there is interstitial fluid outside, and there is plasma inside, right? And we know that capillaries have variable levels of permeability, right? And which of these fluids is going to be higher pressure? Yeah, the plasma is higher pressure and this one's lower pressure. So what moves out is always water and small solutes. What small solutes? Anything big enough to fit through the holes, anything small enough to fit through the holes, like ions, right? Shouldn't be whole big proteins or whole blood cells. That's a bad day when they move through. But water, small solutes, sometimes even nutrients move out. So water and small solutes. Now, you do this at every single capillary in your body. It is where your interstitial fluid actually comes from. It originated as plasma, right? There's a mechanism of turning it over and bringing it back later. You do this really super effectively at your kidneys, because your kidneys have tons of surface area of their capillaries, 1 million nephrons per kidney. In addition, they're super duper permeable, and the pressure is higher, so there's a bigger pressure gradient. Does that make sense? So your kidneys are super duper effective at filtration, but all of your capillaries do filtration. So filtration metaphor that you've run into is making coffee. Far more effective metaphor in Texas than it is here in Utah, but I know you've seen it on TV, or you've been to Starbucks to get a hot chocolate. So this is how the filtration process works. So what you do is you have a membrane that has holes in it, right, which is the coffee filter, right? And then you are going to put stuff on top of it, and then you are going to use pressure. In this instance, you can use the pressure of gravity, but if the pressure was high enough, you could get it to go sideways, right? So you are going to pour water, and the pressure is going to pull anything that is small enough to fit through the holes with the water. So you'll get coffee on the other side, right? But draining noodles, exactly. So, it's not selective about what it's pulling through, it's step by side. So if I put red Kool-Aid in there, I get red Kool-Aid through too. If I put dirt in there, I get dirt, right? It's also not dependent on concentration gradient, because even if there were already coffee grounds in the pot, you would still get stuff moving through. It's only pressure difference that matters, right? Now, what it should do is leave behind the big stuff. But we know that if there's a big enough pressure differential, you can get a bowling ball through a coffee filter, correct? Just only once, right? Can you imagine now what chronic hypertension does to your kidneys? Yes, and you can end up with proteins in your urine, yes? You can then end up with whole red blood cells in your urine. You can then end up with kidney failure with chronic hypertension.
Can kidney stones be that way, or is that?
Kidney stones are formed after the filtration occurs. They're formed at the back end, right? They're formed like after it's already gone over there. Go ahead.
So is it kind of the same way as like the weather system? So it goes from high to low pressure, so the high pressure is inside the blood vessel, and the low pressure is the
exactly. And so it's going to move out. Or at the kidneys, the lower pressure is the urine, by the way. Right. It moves into the urine. It doesn't move into the ureter yet. It moves into the proximal and distal convoluted tubules. Does that sound familiar? And we'll get to the kidneys specifically later. Okay, so do you see why I think this actually, you can talk about it at the same time as you talk about transepithelial transport? Because even though it's not concentration driven, it is moving something across an epithelium, right? So I think it's good to talk about it the first time. So now, let's talk about what this would end up doing. So let's imagine this is your kidney, right? Instead of just any capillary. These are the capillaries of your kidneys. It's going to move water, which is why you pee. It's also going to move small solutes. And importantly, some of the small solutes that it moves are urea and uric acid, nitrogenous waste products. That's part of how your kidneys work, is those are small enough and they get pushed out and then they stay in the urine, they don't get pulled back, really? Okay, so what if your kidneys are failing you? Diabetes is famous for causing kidney failure. Chronic hypertension can also cause kidney failure. So what if your kidneys are failing you? Can you see if your kidneys are failing you, then this might start to, for instance, increase in concentration in your blood plasma to the point that it starts to become problematic? Because where's urea come from? You get it by the catabolism of proteins. Can you stop eating proteins? Not if you want to live, okay? So you're going to get urea building up in your bloodstream, right? I mean, you can change your diet a little bit, but you're not going to be able to completely stop eating protein. So this will build up in your bloodstream. What do you do if your kidneys aren't working? Well, if your kidneys really aren't working, what you can do is a distant plan B, definitely not first choice, would be to do dialysis. So let me teach you how dialysis works. This is hemodialysis, and there are a couple of different kinds, and there's been some big breakthroughs since I learned this part. But I want you to think about what should be happening and then what we're doing here, and I'll teach you how dialysis works. Hemodialysis is specifically cleansing the blood. There's also peritoneal dialysis, which cleanses the peritoneal fluid, and there's some other kinds too. Okay, so here's what's going to happen with hemodialysis. What you're going to do is you're going to have dirty blood, right? Let's concentrate on urea. There's other stuff in there, but let's concentrate on getting urea out of there. And what we are going to do, we're going to take this dirty blood and we're going to run it through a dialysis machine with a little replaceable, disposable chamber in it called a dialysis chamber. I've got one in my office. I forgot to bring it. I'll try to remember to bring it next time. This dialysis chamber, what you're going to have is highly, highly curved tubing running through it, okay? And the tubing will completely separate one side from the next. And this tubing is selectively permeable. So let's look at what's going to happen. What's going to happen is I'm going to put in my blood, goes in that side, and out this side, back to my body, all right? And the problem I have, among others, is that it's got a high concentration of urea in it. Good? Okay, so high concentration of urea. Now, let's look at how you can cleanse it when this fails. When that fails, what you can do is you take what we call dialysis solution. Dialysis solution, which is pretty variable about what's in it. And what we are going to do is in this dialysis machine, we're going to send the dialysis solution in that way and out that way, and then we're going to dump it. We're not going to cycle it through, okay? And why do we do it countercurrent like this? Any idea? Go ahead.
My guess is that as it gets up more, the concentration of urea gets higher. And so if you did low concentration urea to low concentration urea, it's better. And then higher to higher would be easier to go.
It's actually the same as it goes through, but oh yeah, I see what you mean. Go ahead.
Increased surface area connection between the two.
Yeah, so countercurrent exchange, I don't know if you guys learned this in chemistry, but countercurrent exchange actually gets better transport when you run two things against one another with countercurrent exchange. So let's look at what's going to happen. So I will move urea from here to here as long as I make this membrane permeable to urea, yeah? And you can design the membrane so that they're permeable to urea. So urea will move from here to here. And I better make the solution low, I mean 0 concentration of urea, right? That makes sense? So I'm going to put something in that has 0 urea, and I mean this permeable to urea. What?
Right, so then it pushes the urea out.
Yeah. What kind of transport is this actually?
Diffusion.
It's just diffusion, right? It's diffusion. Now, it was supposed to do filtration, but that failed us, so we're going to use diffusion, because diffusion is selective about what moves, right? It's selective via concentration gradient, but it's still selective, right? So it's moving it in, and then what happens is you dump it and continue to dump it so that I always have a super low concentration of urea in the dialysis solution. Now, if I don't want my blood volume to change either greater or lesser, I better also make the dialysis solution blank tonic to my blood plasma, isotonic to my blood plasma, right? Now, can this be manipulated? Yeah, you can do more than just urea if you do a different membrane, right? You can actually change the chambers. You can say, I've assessed your blood plasma and you've got a high concentration of this particular toxin, low concentration. You just change out the chamber, right? Does that make sense? Now, as your kidneys really, really start to fail you, you could have to do this 6 or 7 hours per day. That is not good. It's basically all of your kidney functions are being sort of outsourced. How effective are your kidneys at doing this? Well, your entire blood volume goes through your kidneys every 5 minutes. So they are incredibly effective at filtration when they're functioning properly, and when they're not, you can do this. It is a solution that you can probably do for a while, but it is not a cure. And if you were doing it 7 or 8 hours a day, you're probably on a kidney transplant waiting list, but there are not enough kidneys. So, and you only need one. Your kidneys are kind of redundant. You can actually live with a single kidney as long as you're not doing a lot of shots and a lot of drugs, right? Then you probably can't. Does that make sense? So this is based on diffusion when your filtration didn't work. Good? Okay, so now, with our last few minutes, does anybody have any questions? With our last minutes, we will start with the next set of notes, but it completely corresponds exactly to what we've just been doing. You guys are way quieter than the other class, and I don't know whether that's really good because you're all absolutely independently brilliant and just don't want to share. Is that why it is? Yeah. Okay. Generally speaking, the quieter classes don't do as well.
Hey, wait a second.
It's true. But I'm telling you. No, I'm trying to scare you before you take the test instead of after. So give me some stuff back here. Okay, so this next set of notes is going to depend on this story again, but then we're going to start messing it up. And so my goal for you before I see you guys next time is to make absolutely certain that you understand this whole development of the resting membrane potential. So what we're going to do is we're going to start here where we did last time. And I'm going to add a little bit of detail to it, and I'm going to give you the opportunity to ask as many questions as you want about this process of building the resting membrane potential, because then next time, after we've built it, we are going to start to stick in new kinds of channels, open them and close them, and change it dramatically. So you really have to understand how it got to be this way in order to be able to do that. So let's start there. So this is chapter 7. This is where, by the way, I think we really diverge from 1610. Okay? This is where I think that hardly anybody does this in 1610, but you really have to start with this part of the story. So it gets into the real physiology part now. All right, so start where you know, okay? Ask as many questions as you want to. Make sure that you understand each thing I say before we move on. This is one of those instances in which if you are unsure, ask right then instead of 15 minutes later, because 15 minutes later I just told you 5 other things that depended on that first thing. Okay, so here we go. We're going to get to here again, but we're going to build it slowly, and you're going to ask me questions if you get confused. There is this figure in your textbook that, for those of you who are very visual, this goes through exactly the story that I am going to tell you. This is establishing what we call the resting membrane potential. And I know you can't read it all from there, but for those of you who are visual, what I might do is go through the video, right, and then look at the picture and make sure that you understand each thing. So the question that she asked is like, how the heck does all of this happen? Is it like all at once? What happens first? Well, what happens is that it all happens at once, and this resting membrane potential is established all at once. But I want to separate it out and make sure that you understand each of the parts and add just a little bit of information for you. So if you start out at the beginning like you did now, like you did this time, let's look at the ion composition of the intracellular fluid, add just a little bit of detail that you didn't know before. So the ICF contains a high concentration of potassium, which is of course a positively charged ion. That is where the vast majority of your potassium is sequestered. As a matter of fact, if it all dumps out at the same time, like with burn victims, you are so rude. So potassium kind of needs to stay sequestered in there. It has a relatively lower concentration of sodium. We know that, right? And then, although we didn't talk about them too much before, the primary source of negatives on the inside is these big, huge, negatively charged proteins. Structural proteins, enzymes, cytoskeletal proteins, a bunch of stuff that's in there. And they're not going anywhere on accident. They're big, non-penetrating proteins that are just going to kind of stay there unless you move them on purpose. So those are too big to cross the membrane. And then let's look at the ion composition of the extracellular fluid. The ion composition of the extracellular fluid has high concentration of sodium, low concentration of potassium. I didn't tell you what the primary source of negatives is out there, but now I will. It's got a relatively high concentration of chloride ions. Okay? A bunch of other negatives too, but that's the major one. Okay? So the cell membrane lies between the ECF and the ICF, and that it allows for a concentration gradient for the ions, and eventually it will also allow for an electrical gradient. So what we want to talk about is establishing what we call the resting membrane potential. So stick with me for a second because the terminology gets a little silly. Okay, resting membrane potential. Do you see that once we have gotten to this point, we have generated the potential that any ion could be motivated to move across the membrane? Do you see that? Okay, and it's not just true for sodium and potassium because there is a charge. Now it's true for any ion, right? Any positive ion would be attracted where? In. Any negative ion would be attracted out. That's going to be important, and we use it to our advantage all the time, despite the fact that we generated it just by using sodium and potassium. Okay, so the resting membrane potential, what I want to build one more time and make sure that you actually get it this time is how did we get to the point that we have a charged or polar cell membrane, right? Okay, so do you see that the cell membrane is polarized? Yeah? Okay. So this is polarized. Eventually we will measure how polarized it is and then we'll start to change it. Okay, so at time zero, this is the false but useful story. At time zero, if we can imagine that the ICF starts out electrically neutral and the ECF starts out electrically neutral, meaning that for every positive there is a negative. You with me? Okay. And then I stick in just leak channels. I have not gotten a charge yet, right? I'm telling the beginning of the story. Stick in just leak channels and then acknowledge that initially, of course, the cell membrane is more permeable to which ion? Initially. Potassium initially. So we stick in leak channels. The cell membrane is leaky, but sodium and potassium leak at different rates and in different directions. So potassium always leaks out when given the opportunity, and sodium always leaks in. And before we have a charge, the reason that they are leaking is just due to concentration gradients, right? We haven't built a charge yet. But we said, of course, that potassium is faster initially at leaking out than sodium is at leaking in because there's a greater concentration gradient and more channels. Good. And so what we let it do is, given just a minute and no charge at all, we let it sit there and leak. And then what happens? We accumulate a positive charge where? Outside. We reveal a negative charge inside. And the negative charge, by the way, that we're primarily seeing is these negatively charged proteins. Okay. Note to self, I am not telling you that the entire extracellular fluid is positively charged and the entire intracellular fluid is negatively charged. The charges stay close to the membrane. Anybody know why? They're attracted to one another, like two magnets around a piece of paper, right? So it's primarily the cell membrane that has charge, right? Does that make sense? Okay. So when the membrane has got this polarity, yes, when it has actually achieved this polarity and it's positive on the outside, negative on the inside, about these concentration gradients, you can say the membrane, the cell, is at steady state, or what we call resting membrane potential. But can it rest? It sure as hell can't. What should it do to maintain this for any length of time? Pump its little buns off. For how long? Forever. What will happen to the concentration gradient if you stop pumping for long? Lose it. What happens to the electrical gradient if you lose the concentration gradient? Lose it. Exactly. So in order for the cell to maintain this electrochemical gradient for the rest of its life, you use 40 to 60% of the ATP for every freaking cell that you have, just running the sodium-potassium pumps. And you don't just have one. You don't want me to draw 25, right? You got boatloads of them. And the more often you mess with sodium and potassium, the more pumps you're going to have to actually replace it. Does that make sense? So this is called steady state, or resting membrane potential, which is like the least restful ever, because you have to use your sodium potassium pumps for the rest of your damn life forever for the cell. And this is homeostatic for the cell. It certainly does not mean that we never change it. It means that we always have to put it back, right? Does that make sense? Okay, so one more time. After resting membrane potential has been established, then we can imagine now what it does to the rates at which sodium and potassium diffuse, right? Now that I've got the charge, one more time, which ion speeds up and stays faster for the rest of the life of the cell? Sodium. Which ion slows down and stays slower for the rest of the life of the cell? Potassium. So that is the reason that the pump goes 3 sodiums, 2 potassiums, 1 ATP for the pump. Got it? Because we really only for an instance in the cell's life do not have to deal with electricity. For the rest of the cell's life, you certainly do. Everybody okay? Okay. So the sodium and potassium pump maintains the chemical concentration gradient. One last thing, and then I'm going to let you guys ask questions. The last thing is, what about the proteins? We don't have to worry about them because they're pretty much trapped, right? They're not wiggly. You don't move them on accident. So you can ignore them. It's nice and fizz when you can ignore something, right?
So the proteins are negatively.
They're negatively charged, yes.
So they're kind of pushing because the membrane on the inside of it is also negative, so they're kind of attracting each other, right?
Well, no, the proteins are the primary source of negative charges.
Oh, okay.
Yeah, they're the primary source. I mean, there's other negatives in there. There's like chloride ions in there too, it's just not as high concentration. There's HCO3 minus in there. There's several negative ions in there. Okay. So you can ignore the proteins because they're not going anywhere on accident. What about the chloride? The high concentration of chloride. You can kind of ignore that too, but only because chloride is high concentration on the outside, so it wants to diffuse in, but it also wants to diffuse out towards its opposite charge. And they almost balance each other out, so you can ignore it, kind of like water in an isotonic solution. I mean, it moves, but it doesn't like have any net movement. So for all practical purposes, you can ignore chloride. Unless you've got cystic fibrosis, and then you got some serious problems with your chloride channels. But in most of us, we can ignore chloride. Does that make sense? Okay, so time for questions. Yes?
This might be a dumb one.
It's probably not.
Is it possible for a channel to get clogged by a large protein, or is it just kind of like, or is it just, does everything just kind of keep moving around?
Possible. Yeah, definitely possible. I doubt it would stay there for very long, though. So probably every damn thing that could possibly go wrong with cell physiology has gone wrong with cell physiology, but I wouldn't imagine that the clog would be permanent. There are lots of other forces that are actually acting on the proteins. You've got like water moving by osmosis, you've got charges and hydrogen bonding and all kinds of stuff, moving stuff around, so I wouldn't imagine that would stay. It's not a bad question, though. I never thought of it. What other kinds of questions? Okay.
I didn't catch right there where it says ATP and ADP in that 3-2-1 pump.
Oh, this is just the ratios that the pump runs. But for every time the pump goes, it pumps 3 sodiums out, 2 potassiums in, and uses 1 ATP for the pump, right? And what that does is, after your electrochemical gradient is all set up, it pretty much is going to compensate for the diffusion rate of sodium and potassium. And it has to use ATP in order to do it. Does that make sense?
Yeah.
Okay. Yeah, Charles.
Why do they if it's not so resting, why do they call it resting membrane potential?
Because wait until we start messing with it. And then you're going to feel like this was rest. We're going to mess with it on Wednesday.
So this is basically like the baseline for everything.
Yeah, this is just sitting, I mean, it's just like you sitting around homeostatically maintaining your body temperature. It takes a lot of ATP to sit around and be a mammal, right? So this is the same as, this is what cells are doing at rest, which is not restful.
So nothing's acting upon it. It's just kind of keeping its.
Yeah, it's just sitting there trying to maintain homeostasis despite the fact that like it's got diffusion and other kinds of things working against it.
Alright, because that's the
Okay.
question.
What did you say?
Because cellular respiration.
Because cellular respiration. Where'd all that damn ATP come from? Cellular respiration. What's it trying to accomplish? Homeostasis. Yeah, exactly. You got it? Okay. So, do you really super got it? Okay. One question, and then you. Go ahead.
So the 3-2-1 pump that's
Uh-huh.
happening in that original one is not the same as the facilitated when that's a different protein that brings glucose in with
It's a different protein.
sodium.
You will use this
But
one
it's
to fix
working-
it.
It's working in conjunction with
Yes, exactly. So the sodium potassium pump does
the
this.
concentration goes out there,
Yes.
and then it wants more inside.
Yeah.
And as it comes in through a different protein, it's going to bring some
Yeah.
glucose
So-
and or other things with it.
Yeah, if in addition to this, maintaining homeostasis, we also did a whole bunch of secondary active transport to bring glucose in, what can you predict for me about what this pump would need to do?
Get rid of that sodium.
It'd need to work faster, right? You guys have heard about the red queen, right? You're running as fast as you can just to say, and still, if you want to go anywhere, you have to run faster still, right? So that's what your sodium potassium pump does. That make sense? And as soon as you start to mess with it, it has to go from its baseline level to faster. It can go faster than this.
I have a follow-up.
Okay.
So that's resting, and then as the glucose is coming in secondary, that's when we're moving away from resting and into
Yes, that would be not at rest.
Charles'
Yeah,
work.
that's just doing more than resting, right? Go ahead, your question.
So I just want to make sure, so before it reaches resting potential, so the potassium is going faster
Yes.
out and sodium is going slow, but then once it hits it, then they swap and
Yes,
sodium
exactly.
goes faster.
Uh-huh. Yeah, and you can use the Nernst equation to calculate that, or you can freaking trust me on it if you want to calculate it. The Nernst equation will allow you to calculate based on concentration gradients and based on the electrical nature of the cell, which ion is going to move more quickly. But once you have actually established the charge, then the charge will impact the speed and the concentration. Like I told you guys, charge impacts the speed, the concentration gradient determines direction. Is that okay? Okay. So let me give you a preview of things to come without doing anything really difficult. So this cell membrane is polarized at rest, yeah? Polarized? Okay. There is a way to measure how polarized it is, believe it or not. There are teeny tiny little voltmeters that you could stick in. And what they would do is they would measure the electrical difference between the first fluid they hit, which is the ECF, right? And the second fluid that they hit, which is the ICF, and compare them one to the other. You guys with me? Okay. And what they would do is they would come up with a number. The number would be negative because the first one was what you used as your initial solution, right? And then the second was measured with reference to it. Does that make sense? So it's going to be negative. And then it would have some kind of magnitude. And it actually, on average, measures at minus 70 millivolts. You don't have to know what the hell a millivolt is. Could be a kumquat or an elephant. It doesn't make any difference as long as you stay with it, right? As long as you stay with the same measurement. So that's going to be minus 70 millivolts at rest. That's about the average that all of them are. Cells, all of your cells, have this as homeostatic, positive on the outside, negative on the inside, and they average about this. Some of them are 75, some of them are 60, some of them they average about that. Okay, so just a couple of logic questions and then I will let you guys go. If this is just at rest, now I want you to imagine that I'm going to start to do something else. I'm going to stick in some ion channels and open them. Some gated ion channels and open them. This is in addition to all of this, right? This has just got me to resting. What if I stick in a gated sodium channel and open it? Would that number change? Yes, it would. What if I stick in a gated potassium channel and open it? Would that number change? Yeah. Would it change in the same way? No, it wouldn't. Okay. So what we are going to do is leak channels got us to resting, and now we're going to start opening and closing gated channels that will be stimulated by chemical messengers, neurotransmitters, and those kinds of things. Go ahead.
I was just going to say, the change depends on which way they're moving though, right?
Exactly. Let me see if you guys can tell. Do I have? Okay, I got 2 minutes. I'll tell you guys again next time, but just let me see if you can predict it. Okay, if this is polarized, minus 70 millivolts is polarized, that's how it's polarized. You could get depolarized or hyperpolarized. If you got depolarized, then the number would move closer to 0 because you'd be less different. And if you got hyperpolarized, the number would move further away from 0.
Yes.
Yeah? Okay. Question for you. Opening which channel? A gated sodium or a gated potassium would cause depolarization. Anybody disagree?
Isn't there more of the potassium going around?
It has to do with directionality, not so much concentration.
So,
Go ahead.
Would it be sodium because it's moving faster?
It's not the speed, it's the directionality. Go ahead.
So there's more sodium outside than in, right?
Mm-hmm.
And so if you open up the sodium channel, it would bring it closer to 0 because it's positive, right?
Okay, so if I open up a sodium channel, and you don't have to get this today because you guys are a couple minutes ahead of the other class anyway. I just want to sit and chew on it until Wednesday. Ready? If I open up a gated sodium channel, sodium will do what sodium does, right? Which is try to move in. So if I take positives from the already positive outside and move them toward the negative inside, what's this number going to do? Go closer to 0. Yeah? So they will be less different, less polarized. However, if instead I opened a gated potassium channel, what would the number do? I would take the positives from the already negative inside and put them toward the already positive outside, and what would the number do? It would go from minus 70, not towards 0, but minus 90. Got it? Go ahead.
So you were saying that the concentration dictates the direction, right?
Yes.
And the electrochemical gradient dictates the
Speed. Mm-hmm.
Okay.
Yeah. And that's like truthy. It's true almost all the damn time, and like just in a couple of situations, like with chloride, sometimes it'll cancel out, but they have to be really, really, really close to one another to cancel out. Good? Okay, so that last little part, you don't have to know yet. We're going to go through it immediately at the beginning next time. And we will talk about your test next time too. It's supposed to be on Monday, but I'd kind of like to give you guys until Wednesday just to have absorption time to like chew on it, and maybe have a quiz on Monday.
Are we having a contact quiz on Wednesday?
No, I want to have it no. I want to do it after this. So either it'll be a take-home quiz, or if I move the test, it'll be on Monday, and then you'll have your test on Wednesday. You guys did awesome once you started talking to me. I like that better.